In this lab, we'll begin our studies of the anatomy and physiology of the cell. We'll take a look at the cell membrane, we'll define the cytoplasm, and we'll look at the structure and the function of the non-membranous organelles. One of the diagrams that I'll be using in this presentation is from our lab manual as well as the textbook. This is a diagram of the cell, and what I like about this is it shows you where all the parts are, and then they give you the structure and the function of these parts. If you notice, the ones that are in tan here, all these over here, and this guy up top, these are the non-membranous organelles. This is what we're going to focus on in part 1 of this lab. In part 2, we'll take a look at these blue boxes. These are the membranous organelles. So take a look at the first structure. This is known as the plasma membrane. You could also call it the cell membrane. That's the way I labeled it here. And you could even call it the plasma lemma. Think of the cell membrane as a barrier, right? It's functioning as a barrier that separates all the structures on the inside and the fluids on the inside of the cell from the fluids on the outside of the cell. We call the fluids on the outside the extracellular fluid, and the fluid in here we call the intracellular fluid, or we'll see in a minute the cytosol. This barrier, however, does allow certain substances to, say, move into the cell, and then other substances to move out. We say that the cell membrane is permeable, right? And more specifically, we say it's selectively permeable. So here's a picture from our diagram. You can see here, here's the cell membrane. All of this kind of pinkish color here, reddish pinkish, is the cell membrane. Now, here is a blow up of the anatomy of it. Right above my cursor, that red structure with the two little fibers coming off of it is known as a phospholipid. The building blocks of the cell membrane are these molecules known as phospholipids. The round part is known as the phosphate head, and you'll notice it faces water, it likes water. The two strings hanging down are the fatty acid tails, which don't like water. When something doesn't like water, we say it's hydrophobic. When something does like water, we say it's hydrophilic. So you'll see one string of phospholipids this way, and then another string of phospholipids this way. Again, notice the phosphate heads here are facing the water inside the cell. The phosphate head here are facing the water outside the cell. Look at the fatty acid tails. They don't face water. They face each other. They're hydrophobic. And we kind of know that from our life that fats and oils don't mix with water. So this is known as the phospholipid bilayer. They're not showing it here, but within the phospholipid bilayer there would be some cholesterol. That cholesterol would give the cell some strength. Now, embedded in this sea of phospholipids are these big blue structures right here. These are proteins. So they kind of remind me of these icebergs embedded or just kind of floating in the sea. That's the way this is here. The proteins have different functions. For instance, this one protein has a little tunnel going through it. This is what we call a channel protein. So let's just say there was an ion of sodium here. Sodium could easily pass through this channel and get inside the cell. Right? Other ions could get out. So this would be a channel protein. Other proteins act as carriers that carry larger structures into the cell. Others act as receptors. Others act as anchors. Right? You'll see that they have a bunch of different functions. So the cell membrane is a phospholipid bilayer. Again, think of its job primarily as being a barrier that separates the inside world from the outside world, but it does allow substances to pass in and out. and we say it is selectively permeable. Now, everything within the cell membrane or the plasma membrane, if this is the plasma membrane here, everything on the inside, all the structures within here are within the cytoplasm. We're going to divide the cytoplasm into two parts. The first part is the watery part known as the cytosol. So I labeled just over here, but this clear area where there's nothing in it but water, and it's not a thin slippery water, it's more of a gelatinous water, a jelly-like. This would be the cytosol, right? So all of this here, here, all of this would be the cytosol. Embedded in the cytosol are what we call the organelles. So this pink and white area here, this orange, this black area, these yellow, this yellow and orange here, that's kind of purplish and gray. All of these structures are embedded in the cytosol. These are known as the organelles. We're going to divide the organelles into two types. The non-membranous organelles, right? These are organelles that are just in contact with the cytosol directly. That's the focus of this lab. And then in part two, we'll take a look at the membranous organelles. Those organelles actually have their own membrane around the organelle. So the first non-membranous organelle we do not see on our cell model. You're just going to know this one from the diagram and just know its functions. But if you follow the top of the cell membrane, there are these upward projections here of the cell membrane, this and this. These front ones are cut. We could look into it. But behind them, these ones here are whole. They didn't cut into it. These are known as the microvilli. Micro means small, and villi means finger-like projections. So this cell here has all these finger-like projections sticking up. What that does is that increases the surface area of the cell so that there's more surface area for absorption. That's the main thing I'd like you to know here, that the function of the microvilli is to increase absorption of materials into the cell. So if you think about it, one of the places we would find cells with microvilli would be in the intestines. Right? So all the nutrients that you're absorbing from the diet that you have, hopefully you're eating a nice healthy diet, you're bringing in these nutrients, they're coming in through the microvilli. Right? So they're finger-like projections that aid in absorption. Since they cut them here in this diagram, I just want to point this out. We're going to talk about in a couple minutes these fibers here running through the cell. It's known as the cytoskeleton. But if you take a look here, this is a good picture. The cytoskeleton extends up into the microvilli. These filaments here are actually holding the microvilli up so that they can function better. Here's an actual micrograph. So all of these upward projections, these finger-like projections, are the microvilli. Use your imagination for a minute. If you were to picture your toothbrush at home and you were looking at your toothbrush from the side, doesn't this kind of resemble the bristles of your toothbrush? So some people call the microvilli the brush border. It's just another term we use for the microvilli. The next non-membranous organelle is known as the centrosome. So this whole black area here on this model is known as the centrosome. Then you're going to see two fairly large hollow tubes. Here's one yellow tube running this way, and then one tube running this way. You can notice this middle is hollow. These are known as the centrioles, or we say the centriole pair. If you notice, these are always perpendicular to one another. You'll always see them running at 90 degrees to one another. Here I did a blow up of it, so again you can see the one centriole, and here's the other centriole. So let's talk about it. These yellow lines right here, there are 9 of them. 1, 2, 3, 4, 5, 6, 7, 8, 9. Each one of these is made up of three hollow tubes known as microtubules. We call that a triplet. So this would be one triplet of microtubules, another triplet of microtubules, and like I said, we would have 9 triplets of microtubules that make up the centriole. On our diagram, it's not a black area, it's more of a tan area. So this whole area here would be the centrosome. And then you see the microtubules 1, 2. And again, notice how they're perpendicular to one another. So you can see here in our book, they say we have 9 microtubule triplets, right? So that's the 3 hollow tubes. And again, there's 9 of them. The +0 means this. In the middle, there are no microtubules. All we have are the 9 around nothing. There is something known as the basal body, which anchors cilia and flagella. They're also made up of this type of arrangement where there are 2 microtubules in the middle, so they would call that a 9 plus 2. But for our purposes today, just know that there's 9 triplets arranged in a circle, right? Nothing in the middle. And that's what that 9 plus 0 array is. So the centrioles have a lot of different functions. For this lab number 2 that we're doing, one of the main functions we're going to see is the centrioles are active in cell division. When we're separating the chromosomes to make new cells, the centrioles are going to become important. So we'll see that later in this lab. All right, here's an actual micrograph. So again, you can kind of make out this dark area here. This is the centrosome. And here we're looking into a centriole. Right, this would be the 9 triplets here, hollow in the middle. And then this is running perpendicular, right? See how they run perpendicular? This would be the other centriole. Okay. The next structure we're not going to see on our cell model either in the lab. This one we're going to do more from the diagram. This is known as the cytoskeleton. So I like this picture here. They peeled back the cell membrane and we can look into the cytosol, but you see all these fibers running throughout the cytosol make up the cytoskeleton. One of the main functions, if you think about our skeletal system, our bones, they give us strength and support. And that's exactly what these fibers do. They're giving the inside of the cell strength and support so that the cell doesn't collapse. They even help to kind of increase the consistency of the cytosol. They'll help to hold the organelles in place so that they don't move. They have, again, a couple of other functions we'll talk about more in the lecture class. The 3 filaments or fibers that make up the cytoskeleton, the first one is known as the microfilaments. So this here is showing a microfilament. These little balls right here are made up of a protein called actin. So actin actually makes up this entire filament. If I go back to the slide of the, let's go to this one here, of the microvilli, all of these fibers here are actin, but notice, right, you can see it up here, the actin extends up into the, here's a good picture, up into the microvilli, and they give support so that the microvilli stay standing up. Otherwise, they would collapse and they would be non-functional. Some other fibers we have are intermediate fibers. Again, just think of them as support fibers. And then the biggest one, fiber that's part of the skeletal cytoskeleton, is known as the microtubule. They're made up of these balls, these green balls made called tubulin. These were the tubes that were part of this. In here, there would be 3 of those hollow tubes making up a triplet. So we see the microtubules as part of the centrioles. Right, they have other functions also, but that was one of the ones we had looked at. So all of these, the microfilaments, intermediate filaments, and the microtubules, they collectively make up the cytoskeleton. And like I said before, think of them just like our skeletal system, strength and support. The next two structures, cilia and flagella, we do not see on our cell model either. So these you're going to know more in theory. Extending up from some cells are these short hair-like projections known as cilia. Now, these don't do absorption the way the microvilli do. Instead, when something comes into contact with the cilia can beat, they actually move, and they propel the substance across the cell. As an example, our respiratory system, the respiratory passages, are the cells there are lined by cilia. Many of the cells are lined by cilia. So let's just say hypothetically you breathed in some dust. The dust would get caught on the cilia, and the cilia would start to beat and move that dust back up so that we could cough it out. And so those would be kind of motile cilia. Just be aware there are other cilia that don't beat in the body. These are known as primary cilia, and they're out there more to kind of just sense things. But what we're talking about in this lab are the cilia that do move, and as I've indicated here, they propel the substances along the cell surface. Down over here is a picture of the male sperm cell. So you'll see the male sperm cell has a long tail. That tail is known as the flagella or the flagellum. And the job of the flagella is for that tail to actually whip back and forth to propel the sperm. All right, so these are only found in in humans in the male sperm cell. The female doesn't have any cells that would have flagella. Again, both of these structures you're going to know in theory from this presentation. We do not have it on our cell model. The next structure is an important one. We talk a lot about this in AMP1. These are known as the ribosomes. So all of these white dots, you can see them over here, and I labeled them on the right side. All of the white dots are the ribosomes. If you were to actually look at a diagram of a ribosome, you would see that it's made up of two parts. There's a small part known as the small ribosomal subunit and the large ribosomal subunit. Right? When they come together, they make a functional ribosome. The main thing I'd like you to know about the ribosomes is that they are involved with assembling proteins, right? So they're involved with protein synthesis. Right? The word synthesis means to produce. So whenever ribosomes are in the area, that's what they're doing. They're making proteins. Now, sometimes the ribosomes can be free in the cytosol. Other times, like up here, they're attached to this green structure. Then they're called fixed ribosomes. So you may have heard of something called the endoplasmic reticulum. Here, the ribosomes are attached to the endoplasmic reticulum. So that would be an example of fixed. Again, here's a blow up. And then from our book, over here are some free ribosomes. They're just kind of free in the cytosol. And then over here, there are fixed ones attached to the endoplasmic reticulum. The last structure are these hollow cylinders. We're not going to see this on the, excuse me, we're not going to see those on the cell model. These are kind of like, I think of them as part of the digestive system of the cell. If we have any abnormal or damaged proteins, these structures will actually take those proteins apart and recycle them for us. They are called proteasomes. So whenever you hear the term proteo, think proteins. Right here they actually have the proteolytic enzymes in there. Proteo is proteins. Lytic means to break down. So those are all of our non-membranous organelles. Again, the only two you're going to see on our cell model are the centrosomes with the centrioles and the ribosomes. All the other ones you're going to study from the diagram, and you should know the function of all of just a general function, very, very simple, of all of the organelles.